how biological diversity is generated. What makes the differences between different animals, between different plants, between animals and plants? And you might think, well, evolutionary biology, isn't that a field, a discipline where you go out into the field and you collect animals and plants? Here's a picture of me actually during my Ph.D. in Asia collecting insects that live on this plant. But the part of my Ph.D. that's not shown in this picture from 25 years ago is the part when I was in the laboratory sequencing the DNA of these insects, back when it was actually really hard to sequence DNA. That was the technology component of my thesis. And I've been asked to talk about technology in biology, but I'm actually going to be a little bit narrower and really talk about technology in evolutionary biology, because I think it's kind of an unusual combination. You might think, as I said, what is the role of technology in evolution? But if you think back, actually I would say greater than 90% of the big discoveries, the really important work in evolutionary biology, was driven by technology, going right back to the origins of evolution. People, how did Darwin and Alfred Russel Wallace come up with the idea of evolution by natural selection? Well, they went on long voyages around the world, collected animals and plants in many different continents, and they were taken there by the best technology of the day. These great sailboats that were built by England and shipped natural historians and did a lot of other things, took over lots of countries, caused lots of havoc, but the natural history was fantastic. They brought back remarkable specimens from all over the world, and they started comparing them and thinking about them. How is this diversity generated? Why do I see similar species here? Why do I see some similar species in South Africa and Australia? And it was these comparisons that allowed natural historians like Darwin and Alfred Russel Wallace to conceptualize that these must have come from a common origin, and that there was a mechanism that drove this diversity, and that mechanism is natural selection. So technology has been at the heart of evolutionary biology since its origin, and ever since technology has been driving new discoveries in evolution. So today what I'd like to do is take you to another level, the level that Darwin and Wallace studied, of whole organisms on continents, but the role of genes and cells and neurons in generating the diversity that we see around us. We're not going to have time to touch on all of those topics, but for example, the role of genes, the role of DNA has been elucidated. I hope many of you are familiar with this observation that there are not two great groups of organisms on Earth, what we used to consider the bacteria and the eukaryotes. Through the magic of DNA sequencing in the 1970s and 80s, we now know that there are three major groups of organisms: the archaea, the eubacteria, and the eukaryotes. And that was revealed only by the latest technology today. And this sort of technology, DNA sequencing, has revealed all sorts of magical things about us. For example, most of us carry little bits of Neanderthal genome in our bodies. That was revealed only very recently by sequencing little bits of fossil Neanderthals and comparing them with modern humans. So, DNA sequencing has clearly had an enormous impact on evolution, but there are other technologies that are giving us new insights, and those are the ones I want to talk about today. So, first, let's take a look at what we're going to talk about. I'm going to talk about the evolution of behavior, which is the focus of my laboratory and has been my passion for decades. So we're going to first look at this little movie that we made in the laboratory of a male fruit fly on the right and a female fruit fly in the center. And we're going to see what happens. You know, when a male fruit fly meets a female fruit fly, he has only one thing on his mind, and that is to convince the female to mate with him. And this is what he does to try to convince her. So here he is, he's vibrating his right wing at the moment. She's grooming her head, she's not so interested yet. Grooming her forelegs. Okay, he touches her, and then he swings around in this beautiful sort of dance, and he's still extending the wing and singing. And now he's pumping his abdomen, which is making a little clicking sound that you can't hear. He's doing this little waggle with his wings, and then he's going to do a little scissoring with his wings. and then he vibrates one of these wings. What is going on in this song and dance? It's actually a song and a dance. And we know this because with a different setup, we can actually put these flies on top of a microphone. So the black region behind these flies is now a microphone. The image is lower resolution, but the beauty is we have a microphone so we can hear what's going on. Hopefully these will work. So I hope you could hear that the male was making two sounds actually. A pulse-like sound, type sound with his wings, which he makes by vibrating the wing and stopping it and vibrating it again very rapidly with about a 35 millisecond pause between those parts. And then a hum, which we call sine songs. And the remarkable thing about these songs is that every species sings a different song. Even species that look identical to you and me sing different songs. They're the most rapidly evolving behavior in these animals. And we can illustrate that if we look at the song traces. So Drosophila melanogaster is the one you just saw. That's the workhorse of genetics for the past 100 years. But it has a very closely related species, Drosophila simulans, that sings a similar but slightly different song. And we're going to play these songs so you can hear the differences. I'm going to play these 3-second clips that you can see on the left. The 100 millisecond zoom-ins are so you can see the details of it. Right, so you've heard that it's a slightly higher frequency pitch. Pulse events are slightly higher frequency, which you can see by the narrowing of the song traces. And here's some more divergent species, Drosophila ananasi. And then my favorite species is Drosophila persimilis, because it sounds a little bit like a tree frog. Remarkable diversity of songs. So the question we ask is, how did that happen? How does the genome evolve to generate differences in behavior? That's a big part of the lab, and I'm not going to talk about that today because there's another component, which is, how does the brain evolve to generate these differences? And that's where new technology is really driving breakthroughs in this field for us. And that's the stuff we're going to talk about. So just to emphasize the fact that every species we've tested, these are all different species we've looked at, every species sings a different song. So whatever it is in the brain that's evolving is generating remarkably fast evolution of these songs. And I want you to think for a moment, I study fruit flies because they're experimentally tractable. But it's exactly the same question as asking, what makes us different from chimpanzees? Drosophila melanogaster and simulans diverged about 5 million years ago. Humans and chimpanzees diverged about 5 million years ago. We share many things in common with chimpanzees, but we also differ in many ways. And it's those differences that I'm most interested in. How can you take a stable system like a brain and change it and make something that's also stable but different? Mostly we think about, oh, when I change the brain, I'm going to break something. I'm going to cause mental diseases. I'm going to cause all sorts of things. That's usually what happens when you change the brain. So the great mystery for me is, how do you change it and keep it still working and doing something new and interesting? Very different question, subtly different from the disease question, but complementary. So let's look more closely at what's really happening in these flies. This is just a diagram now of these flies. So there's the female on the top, the male on the bottom. He's extending a wing and singing, producing this song. She's detecting the song by subtle vibrations in her arista, which is this feathery part of her antenna, the tip of her antenna. She's actually detecting the wind that's made by the movement of his wing. The male starts to sing because he's detecting several sensory cues. The approximate size of the female, the fact she's moving around, and also what she tastes like. So the first thing he does when he sees a blob, because they have very poor resolution with their eyes, is he puts out a leg and he tastes it. If it tastes like a female, particularly if it tastes like a virgin female, then he gets interested and he starts singing. You can tell all those things by one quick swipe. Now, how does he do that? Well, his brain, which is his brown part, is integrating that information, multiple sensory cues, and then driving other parts of the brain to drive the muscles to drive this song. The female's brain, on the other hand, that red part, is doing something different. She's trying to decide, do I want to hang around? How long do I want to hang around? So let's look at, we're going to focus only on the male part of this problem for now. We're going to look at the male brain. And the male brain is a complex place. There are hundreds of thousands of neurons in this brain. The remarkable thing about fly brains, which is not true of vertebrate brains, is that they are incredibly stereotypical. So if you identify a single neuron in a fly brain, you will be able to find that neuron in every single other fly. And that means we can name them. And that's what we've done here. So there's a group of about 20 neurons that we call P1. And when I say we, I mean the community of hundreds and thousands of Drosophila neurobiologists around the world. I'm summarizing the work of a large community. At the very end, I'll tell you about the work from my lab. But this community has identified a group of about 20 neurons called the P1 neurons. Their job is to integrate the sensory information and decide, am I going to start courting? And once they start courting, they enter a persistent state, and they keep going. And one of the things they do is they activate one neuron called PIP10. They also activate other neurons, but when they activate that neuron sends a signal down to neurons in the thorax. And those neurons then drive the flight muscles to drive the particular patterns of song. So what do these neurons actually look like? Here's actually a picture of a male brain that's been stained in a very special way to show you those P1 neurons. Those are actually the P1 neurons. Well, how do we do that? How do you image a small subset of neurons out of hundreds of thousands of neurons in a brain? Well, to do that, I'm going to remind you of a little bit of genetics. How many biology teachers do we have in the room? Okay, good. So bear with me, because maybe not everybody is familiar with modern understanding of the way genes work. This is a very simplified version. I think most of you will know that genes produce RNA, which produces protein. That part of the DNA is encoded in what we call exons. So the exons are the gray boxes. Often they're connected, or they're separated rather, by what we call introns. But the RNA is spliced together through some fantastic machinery to produce the full RNA, which is then translated into protein. Right? That's the basic structure of how we think about genes. But that's not enough to make a gene, and in fact, the great revolution in the past 20 years in our understanding of how the genome works is that the most interesting parts of the genome are not in the exons and not in the introns. Well, they sometimes are in the introns, but they're not in the exons. There are these pieces of DNA we call enhancers. Because the job of an enhancer is to turn the gene on and off at particular times in development. It's what puts your head at the anterior of your body and your butt at the tail of your body. If you didn't turn on and off the right genes at the right time, those things wouldn't end up in the right places. So for example, that enhancer will turn the gene on by activating the promoter in one subset of neurons in the fly brain. But a different enhancer nearby will activate this gene in a different subset of neurons. And we know this because we can look at a gene in the genome. It's a big gene called the octopamine receptor 2 from Drosophila. You can cut out pieces of the genome and ask, what does this piece of the genome do? You'll find that this piece drives that pattern, this piece drives that pattern, this piece drives that pattern. What do I mean that it drives this pattern? Well, we can do the following experiment. So we can, there's the original gene again. We can actually cut out that piece of enhancer, that first one, and plug it in next to an artificial system where we've made a little promoter and what we call a reporter gene, something that might glow green, like a green fluorescent protein in the fly brain. And when you then introduce this back into the fly, you generate that pattern that we saw. Now, this is very powerful. We've now labeled, in a very specific way, a small subset of neurons in the fly's brain. We can now exploit this to do something even more interesting, which is we can take this enhancer and drive a special kind of protein called a transcription factor, in this case, GAL4. Now, GAL4 is very interesting because it produces a protein whose job is to bind DNA and then turn on whatever gene it's next to. So we can also put into the fly another gene that has the target sites for the GAL4 protein. And when that protein finds that target site, it will activate any gene of interest. And that gene of interest can be a neurotoxin. It can be a gene that will kill the cell. So you can ask, what happens if I get rid of the activity of that neuron in the brain? We can also do the opposite. We can ask, what happens if I turn on that neuron when it's not supposed to be on? The way we do that, okay, so here again is that pattern. Let's say we were interested in the function of this small subset of neurons in the fly brain. We wanted to find, one way we could do that is to knock out its activity and then try to test lots of behavior and see if anything was disrupted. But a more powerful way to do it might be to turn those genes on at will, turn those neurons on at will. And if we could do that, we could just observe the fly, turn the neurons on and see, does it do anything? Does it drive any particular behavior? And we can do that using another new technology. This was developed only in the past 10 years. Maybe you've heard of this. This is called Optogenetics. The way this works is very simple. I've taken these movies now from the Max Planck Institute, which put these on YouTube. That's their symbol on the top for Germany. It's in German, so I'm going to turn down the volume and then translate, more or less. Paraphrase, more like.
Auge des Menschen.
Unless you know German. So these are now images of channel proteins, proteins that live in the membrane of algae. And when light shines on these proteins, the channel opens up and ions flow through the membrane. And those ions are a signal to the algae that it either wants to move toward the light or away from the light. That's how it's used in nature. But ions flowing through membranes is exactly how neurons work. And so if you can force ions through the membrane of a neuron at will, then maybe you can turn that neuron on by turning light on. And so about 10 years ago, several groups did this experiment by putting these channel proteins from from algae into the membrane of a neuron. And now, when you shine blue light on these, ions flow through the channels. I'm going to do this twice. Ions are flowing through the channels, and when those ions flow through the channels, it induces an action potential in the neuron, causing that neuron to fire, to become active, and turning on that circuit, whatever that neuron is part of in the brain, to drive behavior. All right. So that's been an incredible revolution in neuroscience. And most of the people at the institute where I work, Janelia Research Campus, use these kinds of tools to dissect how circuits work in mice, and rats, and in flies. My lab's a little different. We don't want to know just how these neurons work. We want to know how they evolved. What are they doing differently? So in recent years, we've begun to exploit these technologies to study evolutionary questions by moving the technology that was developed in Drosophila melanogaster into other species of Drosophila. That move, it takes one sentence to say, but it took 5 years to build the tools to be able to make that move. So there's a lot of hidden technology that I'm not going to have time to tell you about. But the end point is, now we can turn neurons on and off at will in these other species. So, what can we do? Let's look again at the P1 neurons. So, what would happen if we just turned on these P1 neurons? So what we're looking at here is a brain of a fly, but not Drosophila melanogaster. This is Drosophila yakuba, one of our favorite species to study. This is very recent data, and the images, I'm sorry, are a little bit low resolution. They are actually taken under infrared light because the channels are not activated by infrared light, they're activated by red light. So what you're not going to see is, in this movie, red light comes on and induces something. So the red light's coming on, opening these channels, starting these neurons to be active, and the following thing happens. Can we bring the lights down, maybe? Okay, so what was maybe a little hard to see Maybe in person.
Okay.
So there's a fly here with the wing extended. Oh,
There we go.
thank you. I'm going to do that again. There's a fly that's singing to nobody. There's no female there. He's singing because we expressed that channel in those neurons and we turned on a red light. But these P1 neurons, I said, can actually drive all of courtship. So we find they actually drive not only that song, but a second song that these species produce, this song. And this kind of backwards walking that it does as well. Not only that, it can also drive the kind of dance that the fly does. It extends its wings, vibrating its wings like we saw in the first movie. In other words, these neurons can drive all of the behaviors of courtship that we've observed in this species. That's very interesting. So that's what the P1 neuron is doing, and that's our understanding from Drosophila melanogaster. But these species actually differ in many aspects of courtship, and also the song. So Drosophila jacoba is on top, Santomea is in the bottom. I'm going to play you these two songs. There's jacoba. Santomea, a little bit faster. Right? These species diverged only 500,000 years ago. Drosophila jacoba lives in the Congo, on the west coast of Africa. Santomea lives on a little island off the west coast of Africa called Sao Tome, only up in the highlands, up in the mountains. These species diverged very rapidly, very recently, and have diverged mainly in their courtship behavior, and also in some other aspects of anatomy. Now, we'd like to know what causes this difference in behavior. It's a nice, simple, straightforward difference. You know, when we study evolution, when we study anything in biology, actually probably any science, we always make philosophical trade-offs. We don't study what might be the most interesting thing. The difference between chimpanzees and humans, I'd really like to know what drives those behavior differences. But you make a trade-off when you're doing science, and you pick experimentally tractable systems. And particularly systems where the differences you're looking for are relatively simple. That's how we get traction. So what we're going to do is ask which part of the brain is involved in generating that species difference. So I told you that PIP10 is the neuron that's driving this circuitry in the thorax. So a very obvious first question is, do the neurons that drive the species difference reside in the brain, upstream of PIP10, or in the thorax, downstream of PIP10? If we were able to turn on PIP10 in both species, we could resolve that question. If the two animals sing the same way, then the differences are upstream of PIP10. If they sing differently, then they're downstream. And this will allow us to start parsing out where in the brain these differences occur. So we've done that. These data are 2 weeks old, so you're getting the real cutting edge of this science. None of this stuff is published. You're the first audience to hear about it. So here is the Drosophila jacoba fly, where we've labeled the PIP10 neuron. There are 2 of them, one on either side. That's where the nucleus resides, in that green dot. There's a neuron that goes all the way down to the thorax. And when we turn on this neuron with red light, this is what happens. It will keep going as long as the red light is on until it dies. It will just keep going. So the average time between these pulse events is about 98 milliseconds. Remember, about 100 milliseconds. When we do the same experiment in Drosophila santomea, again, turn the red light on, much faster. Average interval of about 67 milliseconds. That is, this experiment recapitulates the species difference. That is, whatever is generating the species difference in behavior resides in the thorax of the fly, not in their brain. It's in the ventral, what we call the ventral nervous system. So these are the ways that we're going about trying to dissect where in the nervous system these behavioral differences reside, and that's the future. for us. So I want to now tell you just again where I work. If you're interested in the development of these sorts of technologies and how they're applied to the study of behavior and neuroscience, you can visit the Janelia.org website. There's a lot of fascinating science described there. In addition, my lab's website is also there. In addition, you may have noticed that I have a somewhat different approach to giving science talks than many scientists. In fact, when I give science talks, they're much like the talk I gave to you. In plain language, avoiding terminology as much as possible, no words on my slides, heavy use of black slides so that you look at me, you're not looking at the slide. And I think this is really going to help scientists communicate with one another. But there may also be lessons in here for teachers. Bullet lists are not the answer to grabbing a student's attention, for example. So if you're interested in this topic, I have some very blunt advice on my website called howtogiveatalk.com. It's also howtogiveatalk.org. So the only important part is how to give a talk. Now, I have brought with me a little bit of the technology that we use. One of our portable song recording devices. So I brought some technology and a great technologist. So this is Elizabeth Kim, who's a technician in my laboratory at Janelia. And she has brought out one of our mini song recording rigs. So you can bring this up on screen in a moment. So it's a little box, a little plasticine box. There are microphones underneath. So she put a male and a female fruit fly of 2 different species in here some time ago, but there's a divider that separated these 2 flies until a minute ago, and she just opened that divider. And so now there's the male and the female are getting to know each other. And soon, the males will start singing. Hopefully.
Too many people watching.
It's anxiety.
Does it work? Oh, there we go. I heard it. So she's switching between the channels. Very weak. Okay. Which channel are you on? Yeah, it's number 4, because the male is extending a wing. Okay, so this is technology. It doesn't always cutting edge. You can hear it, right? Yeah, so you can hear it. You can hear it here. All right. So while they're going, maybe we'll hear more in the background, and I'm happy to take any questions you might have. I think we have a few minutes for questions. Yeah. Thank you.